Hi again, back to wrap up with these last couple of videos, our chapter, multiple weeks on respiration, ventilation, gas exchange, all those kind of cool things. This particular video is about the control of the respiratory system. And this should feel like a pretty natural progression. We started with some structure and anatomy, we got into some function, ventilation, gas exchange. Now we bring it all together and determine how respiration, ventilation are controlled in our bodies. Because obviously they have to be like all these autonomic systems. And respiration is one of the most interesting because I think, as I've mentioned before, we can both unconsciously and consciously control it. We can override the built-in intrinsic system that's regulating our rate of ventilation and depth of breath with conscious control, something we can't do with many other elements of the autonomic nervous system. So you can already anticipate things are going to get a little more complicated in that there's multiple pathways through which this control can occur. And I'll try and focus on the ones I think are most essential to know a little more detail about. But the big picture here is that we have a multifaceted, multi-layered control system for respiration that really is quite unique in the body and physiology in general. I'm going to break it into two parts. One is arguably mechanistic still, in the sense that it has to do with matching the pulmonary ventilation with the pulmonary circulation. So obviously, to maximize gas transfer, we want to be bringing the blood supply not only close to the alveoli to allow gas exchange, but also to make sure that the flow through the pulmonary circulation matches the airflow, the ventilation that we see in the nearby alveoli. Otherwise, we potentially, waste capacity or miss picking up oxygen that we otherwise could. So this is this matching, and this overlaps into the control side of things because we'll bring up some control elements in that context. So it sort of fits in this chapter. So the first thing to anticipate is that our perfusion of our lungs will not be uniform. As we stand upright, for example, gravity clearly plays a role, and it will increase or decrease the hydrostatic pressure depending which way you go and whether you're standing on your feet or your hands. But normally standing upright, we have an increased hydrostatic pressure in the lower part of our lungs compared to the upper part of our lungs. And this matters. This is enough of a distance, and blood is dense enough that resulting pressure change is something that affects the amount of, in this case, perfusion that we have going through different parts of the lung. And now the question is, how does that perfusion match the ventilation? Because we also can anticipate we don't ventilate uniformly, at least when we're breathing at rest and taking sort of, slower, more relaxed breaths. When we're exercising, the rate of ventilation goes up, the depth of ventilation goes up, in the sense we take bigger breaths, and with bigger breaths, we get more uniform or more complete ventilation of our lungs. And so it should make sense that we have some kind of regulation to match those two so that we get good, clear transfer of gas, ultimately effective, efficient transfer of gas. Now this figure here shows the situation broken down at least in three regions, and there are three different pressures we have to keep track of here. So we have the alveolar pressure, the P with a capital A, and then we have the two lowercase pressures, the arterial and the venous pressures, which are on either side of the pulmonary circulation. And obviously, as with all circulatory systems, one of the big drivers of flow is the pressure difference across a length of vessel, or length of pipe, if you will, if you want to generalize. So when, as always, we have an increase in arterial pressure over the venous pressure, which we typically have, then one can expect there to be flow. There's at least a drive for that flow to occur. And as usual in these relationships, we have a flow rate as the output and the product of a drive, in this case, the pressure difference, and a permeability or over the resistance, depending on how we formulate this. Again, this you've been seeing all semester long. So we have to then evaluate, when it comes to the actual rate of blood flow into various parts of our lungs, what are the various conditions of pressure and how would they affect that flow? And again, they're affected in two ways. One way is obvious. If the arterial pressure is larger than the venous pressure, as I said, it will produce a positive gradient that will tend to push the blood through that segment of the pulmonary circulation. But when we have alveolar pressure, that pressure you can imagine surrounding the capillaries in the pulmonary circulation. And so if that pressure starts to increase, it's literally going to squeeze down the capillaries, increase their resistance, and reduce the overall flow. So again, this is all driven by pressures, but the consequence on flow you can see is broken out into pressure differences, the drive, and pressure differences between inside and outside the vessel, which ultimately reduce down to a constriction or squeezing of the capillaries in that pulmonary circulation. So these three regions that you see in the diagram, you see the apex, that's the top of the lungs, and there you see a situation where the pressure from gravity is at its lowest. And so overall the pressures in the vasculature and the capillaries are also going to be at their lowest, both the arterial and the venous pressure. And so we can get a situation there where the alveolar pressure, especially if we're breathing somewhat deeply and we have air ventilation flowing in those alveoli, that the alveolar pressure exceeds both the arterial and the venous pressure in the capillaries. And in that case, and you can see it depicted in the little boxes at the apex at the top of this diagram, that you see that there's a restriction in that vessel, literally in that capillary, and hence an increase in resistance and a reduction in flow. So we're going to have lower flow in the apex of the lungs relative to regions further down. The next one of which we see the situation where the arterial pressure now has exceeded the alveolar pressure. And so there we get, some constriction depending on where we are in that region, maybe some constriction, but also a situation sort of in the middle, if you will, where the vessel really isn't compromised at all, where the arterial pressure is enough to withstand any pressure differences, or the pressures are even between at least some segments of the vasculature and the alveoli. And there we see a straight pipe going through. And then we even see a situation as we get closer and closer to the base of the lungs where that arterial pressure starts to exceed the alveolar pressure. Again, because we're moving down, gravity is adding to those pressures, and overall we're going to get an increase in the arterial pressure exceeding the alveolar pressure, which is still lower than the venous pressure. And in those circumstances, we get an increase in flow, a gradual increase in flow as we go down. And at the right-hand side of this figure, you can see the blood flow as a function of this movement from the apex to the base of the heart. And we see that red line shows that the blood flow increases as we go through that middle range until we get down to the bottom, in which case both the arterial and the venous pressure exceed the alveolar pressure. And so along the entire length of those capillaries, there's literally a bulging because there's more pressure inside than outside. And there we get a further increase in the blood flow because we've literally opened the vessels further than they would be under those other conditions. So it's this careful balance between these factors that determines the perfusion, so this is the blood flow, the perfusion through these different regions of the lung. And naturally, if we stand on our heads or stand on our hands, this all flips and we get a flow that goes the other way, where we get decreased flow at the base and increased flow at the apex. And if we're supine, there's not much of a difference at all. So this is, again, mostly gravity driven. So think of gravity in those kind of terms. So that provides a sort of big picture situation of both the venous or the arterial venous flow, the vascular flow in the capillaries through the pulmonary circulation and the ventilation. Now, there's also a local mechanism. Just like there was a local mechanism when we remember way back when we talked about cardiovascular flow or vascular flow, there's some local regulation of the vasculature in general. And here we have also some local adjustments of flow and of ventilation that try and achieve a matching of the ventricular perfusion and ventilation, or ventilation perfusion, this V over Q. This ratio of ventilation to perfusion is something that at rest sits around 0.8. So, ballpark, it's about equal, a little less than equal, let's say. And that matching, it's just what we measure when we actually take measurements. And these are measured in terms of flow rates. So, ventilation is air flow per unit time, and perfusion is the blood flow per unit time. So they're, again, sort of equal, but a little smaller than one if you get really specific about it. Now we have a control system. And as always with control systems and feedback loops, there are a number of elements to that system, as we've described in other parts of the physiology. And each feedback system arguably has a goal. There's some purpose to this feedback system. In this case, it's to match the two, the ventilation and the perfusion, in such a way that we optimize or maximize the gas transfer. That should be clear. The process by which we carry that out is that we regulate the flow, we alter the flow in order to match the ventilation. So this could go either way. We could argue that ventilation gets adjusted in some local way, but that's not what we think happens. We think a lot more of this local response, or if not entirely this local response, is a change in the perfusion of that region. So the perfusion gets matched to the ventilation, and that can be controlled locally through sensors. Every feedback system has to have a sensor. And the sensor at this case is again, very local. It's at the tissue levels, and it's able to sense, and the mechanism is not entirely clear, able to sense hypoxia, or oxygen concentration, oxygen partial pressure that we have. And so the mechanism, we'll get to in a sec, the actuators, once there is a signal, so once there's a difference between a set point and a measured value, as with all feedback systems, there has to be a means to close that loop, to actually actuate or bring about a change in the perfusion to regain this balance, to reduce this difference between the threshold and the actual value. And there we have basically a vasoconstriction or vasodilation mechanism, just as we've seen throughout the circulatory system. And the mechanism by which this works, we think, is a local change in hypoxia, which is sensed somehow by the nearby arterioles. And that's important too. The capillaries themselves have no smooth muscle and no ability to regulate, but the arterioles, sort of one generation up, they have that smooth muscle. So that's where control is likely to occur. It's the only place it can occur. And it turns out that there's enough proximity between the sensors that notice hypoxic changes in that local scale and the smooth muscle controllers that would allow us to vasodilate or vasoconstrict according to that any imbalance that arises. The exact mechanism for this apparently is still unclear. As with many things in the body, another great research project for somebody, but at the end of the day, it does alter the cross-sectional area through vasoconstriction, changes resistance, that changes flow, and the system is restored back to a desired balance is 0.8. This balance between the ventilation and the ventilation and the perfusion in that region. So it's kind of a nice example of a local mechanism. Doesn't involve the brain, but still is important for maintenance of respiration at a very fine scale. And you can picture how this can play out all over the lungs and the pulmonary circulation and adjust things. And it's just, again, one of a number of mechanisms, many of which we don't have time to get into, that allow this matching to occur. Now, seen at various scales, we can have defects in this. It's possible to have mismatches, of course, that maybe aren't quite controlled, especially in the conditions of pathologies, of diseases that arise. And in this figure, you see, again, some examples of these defects and how they might arise. And so the top figure is the sort of normal case where everything is good. The case we hopefully have all right now as we're sitting there breathing. Then below we have these various abnormalities. Now, one of them is the dead space. So dead space, we've said before, is space that doesn't get ventilated for one reason or another, or is not involved in gas exchange. So we spend a lot of time talking about different types of dead space. Some of them are just the spaces in the bronchi. Those are physiologically dead spaces because that air can't be involved in gas exchange. And so anything that increases the local dead space, so let's say we have a blockage in the bronchi or the smaller divisions of the bronchioles going into the alveoli, that would create dead space, literally space where there's supposed to be air going back and forth that isn't. And that's a situation where, of course, we have a terrible mismatch. We have air not doing anything, blood flowing by, never picking up or dropping off any gases. That's not a good situation. In the second case here, you see, if you look carefully, that compared to the normal case, the vessel is a little constricted. So this would be the situation where we have a reduced blood flow, reduced perfusion through that region with normal ventilation. And so we get an altered ventilation to perfusion rate, an elevated one. And that can happen mostly, again, through some occlusion or some blockage in the perfusion bed of that part of the lungs. So you can picture pulmonary embolism, for example, causing either reduction or even a complete block in the flow through those vessels. You can have the other situation where there's a reduction in the ventilation. So this third case where you see the alveolus looking quite small for some reason, or the flow rate going into the alveolus being smaller. So that kind of constriction can reduce the ratio of ventilation to perfusion and also cause a reduction or cause a mismatch, right? And that, again, you can imagine we've talked about those kind of situations a little bit before. And then we can have a right to left shunt. So this is a situation where the blood literally just bypasses that part of the alveoli. So somewhere upstream from the capillaries, for example, there might be a shunt. This can go all the way back to the heart, left-right ventricular shunt. So anything that causes the blood to bypass that part of the circulation that goes right by the alveoli, that can cause this shunt, and the shunt, of course, results in no gas exchange happening at all. So we can have lots of variations of these different themes, different balances between the ventilation and perfusion, and the regulatory mechanisms will be constantly trying to make up for these defects and deal with them locally, quickly, and we'll see shortly even more globally. But this mismatch is mostly controlled by local mechanisms. So now we get to the bigger picture of control of the whole respiratory system, so the entire control of respiration. And as usual, we have a feedback loop here involved. And as usual, or as always with feedbacks, we have a goal. And the goal here, the high-level goal, is to maintain sufficient ventilation with minimal energy. That's another constraint we can add in. It's always effective or efficient when the body minimizes the energy need for the actual benefit involved. And the process steps that we have to monitor ultimately, and the sensors we have to capture, and the actuators, they have to do with the ventilation mechanics that you've heard about before, and to some extent the aerodynamics, the way air, for example, flows in and out of vessels. The points of regulation that we have are, again, the obvious ones: how deeply we breathe, how frequently we breathe, the musculature that controls that, which, as I think I've mentioned before, is very finely tuned. We have a lot of muscles and a lot of nerves that are responsible for ventilation, the mechanics of ventilation we've already talked about. So there's a great deal of control of respiration, even unconscious control. And certainly I think we can even sense when we think about our breathing how finely we can regulate that breathing in practice. A key part of this is the sensors, of course. So how does the body know when there's a need to breathe more, breathe less? And there are two basic groups of sensors. There's one central and one remote. And then there are two gases that are going to control this: the oxygen, of course, and also the CO2. So both those gases we'll see in a sec are very important, and they actually regulate differently in different sensors. So this is where the kind of complexity comes in. But at the end of the day, the goal is to, as usual, create a balance and maintain adequate ventilation. And then we have the feedback loop itself, and the actuators for that feedback loop, that goes through the central nervous system. That's where the control is. And then there's very extensive, as I've said, control through innervation of the various parts of the respiratory system. So here's a picture. So just to remind you, you've seen this one before, just of the even vaguely, delineated here, these various nerves that control the lungs at different levels. You see The phrenic nerve is especially important, and you see that a great deal of that phrenic nerve control also goes all the way down to the diaphragm. You can't quite see that in this figure, but there's a nerve pathway that goes to the diaphragm through the phrenic nerve. You can also imagine how damage at various levels of the spinal cord can have different effects on respiration. So, if we have, let's say, unfortunate break in the spinal cord, the lower we are down, the fewer of these pathways to respiration are involved. And as we move up further and further, by the time we get to the cervical vertebrae, we're really in serious situations. So you can imagine, for example, if the cervical vertebrae were all intact, or the nerves that leave those vertebrae were all intact, then we would still have diaphragmatic breathing available, but not as effective a ventilation by moving the rib cage. So that form of breathing would be reduced, whereas the diaphragm would still be functioning normally. And so this is where this wiring plays a very strong functional role. So now we have to talk about receptors, and there are two families, a central and a remote, and the central ones are the ones that mostly are there to sense CO2. And as we'll see, CO2 is a major control mechanism for respiration. If there's a bottom line to this whole thing, it's that CO2 is the dominant driver, especially when we're in small variations away from our normal breathing. And we'll get to that in a little more detail shortly. But the interesting part here is just think about the sensors. So these are central sensors. They're in the brain. And inevitably, when we have something central like this, we have to think about how chemicals or ions cross the brain-blood barrier. And here's another perfect case, in fact. It turns out that these sensors are mostly sensitive to pH, so sensitive to hydrogen ions. However, hydrogen ions don't pass easily across this blood-brain barrier. So what do we do then, or what happens then? What happens then is that the CO2 can cross the blood-brain barrier easily. And we've seen before there's a hopefully familiar now reaction that involves CO2 forming a weak acid, which then further breaks up into positive hydrogen ions and bicarb. And so that reaction is what ultimately allows us to measure the amount of pH that's present, because the CO2 concentration will be altered by the increase in hydrogen ions, because this reaction, of course, can be driven in both directions. So hydrogen, again, passes poorly. You can see that in this diagram here, especially the lower part of it. You can see that the hydrogen ions are sort of bounced away when they try and leave the vasculature, which is where this reaction initially starts. But the CO2 concentration can transfer easily and can get into these chemoreceptors that are in the interstitial space, nearby, near the vessels. And so again, it's mostly pH sensitive, but pH encoded almost in the CO2 concentration. And this system, this sensitivity to the CO2 levels and ultimately the pH, is something that also changes over the longer time. So this is another factor in these regulatory systems we've seen again and again. There are short-term regulatory mechanisms and there are longer-term regulatory mechanisms. And in the short term, we get a pretty predictable relationship that fluctuates even within a breath. So these are very fine gradations that we're sensing. And then there are longer-term sensitivities that change that will affect things like how we adapt to altitude. And we'll come back to that in the next lecture. And so this really is a powerful mechanism that stimulates us to breathe. So when we think about what happens when we hold our breaths and why we would need to start breathing or could not hold our breaths any longer, CO2 probably plays the dominant role, we think. Now, those are central chemoreceptors. There are also peripheral chemoreceptors, and they are primarily sensors for oxygen, but are regulated by the concentration of CO2. So it's quite an interesting system here. So they're, like I say, we think of them directly sensitive to O2 concentration, but indirectly sensitive to PCO2 and even a little bit to pH. So, and they're located in the carotid sinus. They're near the baroreceptors, same sort of regions, the aortic arch. So a lot of the same places as the pressure sensitives live, the baroreceptors also live these chemoreceptors. But again, they respond to difference in concentrations, not in terms of pressures. this is really where all the oxygen sensing happens. So when we think of hypoxia, anything that reduces the amount of oxygen we have available to us, the sensing for that is almost entirely in these peripheral chemosensors, or so we think. And there are some differences between the sensing of both of them. The key thing here I want to illustrate, and you can see this in this little plot in the bottom here, is that the sensitivity, so the rate of ventilation, for example, which you see in the y-axis, is sensitive to arterial oxygen concentration, which you see on the y-axis, or the partial pressure of O2. But what you see is the curve is flat over a pretty broad range of partial pressures, and only when it gets sort of into the range, turns out around the range of 60 millimeters of mercury, which is quite a bit less than 150 that we would see in inhaled air, or 100 that we would see in the blood. Only when the partial pressure drops down to that 60 range do we start to see this rapid increase in ventilation rates. So this is not a sensor that's going to be so sensitive to those fine-tuning aspects of respiration, because in that range of 100 to 60, there's really not much stimulation that's going to come from this sensor. In contrast to the CO2 sensing, which is very sensitive to small fluctuations in the partial pressure of carbon dioxide. The other factor that you can see in that plot is that there's ultimately a family of plots. We see here just two examples, one that's at 40 or 41 millimeter mercury of partial pressure of CO2, and the other one at 52, so elevated PCO2 levels. And we see here that when the PCO2 level is elevated, that means more carbon dioxide is being, let's say, produced by the body, picked up by the vessels. So what we also see here is, in a situation with elevated partial pressure of carbon dioxide, the upper curve, that we also see for the same arterial partial pressures of oxygen, an increased value of respiration. So the presence of elevated CO2 sensitizes or increases the sensitivity of this O2 sensor and delivers higher inspiration or ventilation rates as a function of the partial pressure of CO2. So this is how the interaction, you can picture the interaction between the partial pressure of oxygen, partial pressure of CO2, both coming together here to determine ventilation rates. This picture shows some of the details. It's kind of fun to go through this, just the details of the mechanism by which this glomus cell, it's called, in the carotid body would sense the O2 concentration and cause a change in an ion channel, which increases some calcium, and that calcium increases the release of a neurotransmitter, and then the neurotransmitter interacts with a receptor, and that receptor generates the ultimate signals. Again, the details here are not so important. Just kind of get a sense of this is a typical almost way that sensors of all sorts respond. They have some sort of initial response, often a second messenger involved, ultimately change the concentration or change the permeability of an ion channel. That increases the concentration of something, an ion, and that causes some release. So this sort of chain reaction here is not an unusual thing to see in our regulatory and the sensing parts of our regulation systems. So it's quite hopefully useful generically to think about this. Now let's see. In this picture, for example, we see what's the overall CO2 control. Because again, I've mentioned there's a direct control via the central sensors. That's what you see on the left-hand side of this figure. The CO2 crossing the blood-brain barrier, getting into the cerebral spinal fluid. There it interacts with these chemoreceptors that I said exist in the extracellular, extravascular space. And then that causes a signal to the brain. And there we see this respiratory control centers. We'll talk about that in a sec too. And that would either cause an up or down regulation of ventilation depending on which way the CO2 concentration changes. And then we see this external mechanism in the peripheral chemoreceptors, sort of indirectly enhancing the sensitivity to oxygen concentration. So that also causes, in the end, through this totally separate pathway, another input into this respiratory control center that would adjust ventilation. So again, this we've seen examples of this before, multiple systems feeding into a central control system that ultimately determines what really goes on. Here we see the kind of the big picture of that central control system as an overview. And there we see not just the CO2 pathway, which we just saw in detail, but also the O2 and even the pH pathway. So the oxygen, again, goes through the carotid aortic chemoreceptors, feeds into the system that way. You have this central mechanism for CO2, you see that. And then you even have this external voluntary, or sometimes sort of quasi-voluntary, this emotional response mechanism, for example. It turns out, I think as we'll see in a second, that we have a natural tendency to take a quick breath before we start speaking. This is not something that we're terribly conscious or even aware of. We certainly don't set out to do it that way. But the minute that mental plan arises to start speaking, there's also an initiation of a quick breath. And obviously, if we're scared or there's some strong emotion, then our breathing will be altered. So this, again, a complicated system. There's all these conscious, unconscious factors feeding into this central control system, which then can act through a number of different mechanisms that you see here to control the rate and the depth of ventilation that ultimately controls how we breathe. And those can, again, they can direct nervous control of the diaphragm to the intercostal muscles throughout this very finely tuned system we have for controlling respiration. So it's really quite complicated and interesting how much variability there is. This is just kind of a fun example of the control of ventilation in action. And what we see here is in the bottom, the tidal volume. You hopefully remember that quantity. So that's just the low level sort of at rest breathing that we do over time. So we have an inspiratory phase of that, an passive expiration phase of that, and you can see a couple of breaths down below here. What you see above is the number of active inspiratory neurons that are firing. So we have neurons that sense that inspiration, and their firing rate goes up as we breathe in. And then that inspiration drive turns off. We reach the end of our breath, and again, there's mechanisms to control when that happens. And then that feedback, those firing rates of those particular neurons, they go back to a very low level, and then it repeats itself. And so there's this ongoing set of stimuli that are driving these muscles, and it tends to be the inspiratory side of this that matters the most. Now, of course, this has a certain pattern of activity. You saw it there in the nerve action firing. What we see in this figure is a series of time plots describing different levels of this system, different patterns that are generated to respond to the drive for respiration. At the top, we see the phrenic nerve activity. So this would be the mean value over multiple recordings, of course, but this is for one inspiratory element of a breath. So we see an increase in activity over time. Corresponding to that, we see a decreasing pressure, interpulmonary pressure. Those are mirror images of each other, as we would expect. And then below that, we see the phrenic nerve activity under a range of different levels of partial pressure of CO2. And we see that as the partial pressure of CO2 goes up, the phrenic nerve activity also goes up. So each breath is going to respond to the PCO2, the level of CO2 in the alveolar CO2, and show an increasing level of activity, and hence drive up the phrenic nerve overall activity, drive down the pressure, deepen respiration, that sort of thing. So, and then at the bottom we see something also we haven't talked much about, and that's the activity from the pulmonary stretch receptor. Now, we again have sort of a small breath version in red and then a larger breath version in blue over top of that. And we see a dramatic increase in the activity from that pulmonary stretch receptor as we have a deeper breath. So this would be the situation that high partial pressures of CO2 would lead to increased respiration, increased activity from that stretch receptor, and that would then cause a change in the phrenic nerve activity. So these are all sort of playing out, in a kind of complex dance with multiple inputs, with a central control system managing the drives that would come from our breathing under these range of different conditions. We have a pacemaker like we do in the heart. We don't actually understand fully why we have that pacemaker, how it works, which is kind of fun to think about. We have a regular rhythm of breathing. That rate of breathing is a function of the previous breath. So if we take a deep breath at one moment, then maybe we'll breathe more slowly going after that because we have an excess, if you will, of O2 available, or have removed an excess of CO2. And all these factors are really playing out the same. The durations, as I mentioned I think before, and we've seen it in those sinusoidal plots, the inspiration, expiration durations are roughly the same. We tend, when we're breathing in a relaxed way, they tend to be the same. And our breathing is continuous when we're awake. You will see when we go to sleep, that actually changes. That's kind of interesting. Most of the control is happening to encourage us to inspire. So expiration is mostly a relaxed activity. Unless we're really forcing it for some, again, conscious reason. In general, the brain activity of the autonomic system is happening, and the sensor feedback or instigation of this is mostly happening during the inspiratory phase, which is what that plot showed before. That's where all the nerve activity is, and then after that tends to be kind of a passive relaxation that occurs after that. Now, when we go to sleep, things change. Lots of things change, and sleep's important. One of the things that changes is our breathing is not continuous. So we tend to have what's called episodic breathing. So, and if you sleep with an intimate partner, or watch somebody nearby sleeping and paid attention, you'll notice this. You'll see the breathing is not always continuous. There's periods where it is. There's periods where it literally stops. It's almost like a breath hold is going on, then it picks up again. The extreme case of this, of course, is sleep apnea. And this is a pathology, and it can become such a problem that every time the breathing stops, we get into sort of dangerous levels of hypoxia, dangerous levels of CO2 concentration, enough that the sort of gasping that follows when the brain catches up is enough to wake the person up. And this is the incredibly disruptive effect of severe sleep apnea, is we don't get good sleep because we're constantly being woken up again by our own breathing. And as we see, this becomes even more common, more frequent when we're at altitude, when that stress of altitude comes into it. So this is, again, not so surprising that the central mechanisms sort of turn down a little bit as part of our sleeping process. And so the control then becomes driven much more by the peripheral chemoreceptors. And we said before, peripheral chemoreceptors don't do a lot until the oxygen levels get substantially lower than normal, get down to that 60 millimeters of mercury partial pressure range. And so this partially explains why it takes a while for that mechanism to kick in and cause us to start breathing again. And we think that explains this typical episodic breathing that we have at sleep. The final, I guess, couple of points I'll make here is that besides everything else you've heard about, there are even more stimulation elements to our breathing that can affect how we ventilate. One of them, for example, is when we sense dust particles. So if we breathe something in, we've all experienced this, I assume, that you get in a dusty setting and all of a sudden you just feel compelled to cough. And that's, again, an irritant response. So the lungs sense those irritants, and that causes a bronchoconstriction or bronchiole constriction, and leads ultimately to the coughing that helps expel some of that dust. So while that's playing out, there's an emotional component to this. Again, sympathetic stimulation can alter our breathing in ways we, just again, because we're excited, because we're afraid, for those sorts of reasons. Temperature can change the sensitivity of these sensors and affect the way we breathe. And as I mentioned before we start to speak, we tend to inhale. We tend to take a breath before we speak, which is very handy. I would encourage you all to take a deep breath before you speak so that you don't run out of air like I'm about to with this overly long sentence. Finally, we also have conscious control, as I've mentioned several times. Now, as we'll see in the lab, we're going to try and stimulate the CO2 and the O2 systems differently. So we're going to stimulate what's called hypercapnia, that's elevated CO2 system, and then we're going to isolate it from that, stimulate the hypoxia system, and try and get a sense of how the body responds to those two separate stimuli. Now, this takes some artificial controls. We'll get into that in the lab. In our normal daily life, both these systems are working all the time. So they're overlapping on top of each other. And of course, it's impossible to say, am I breathing because I am getting a little bit hypoxic, or am I breathing because I've built up too much CO2? And we really can't separate the two in normal activity, and it's not important that we do. In the lab, we can actually delve into that question a little bit more and see which of these two systems ends up being the more powerful driver, and how it generally affects even other aspects of our bodies. So we'll get to that in the lab. It's a really fun lab to do. So with that, hopefully you got a sense of, on the one hand, how ridiculously complicated this is, but hopefully a couple of key elements have emerged. The notion of these chemosensors, the fact that they exist, a little bit about how they function, how one is sensitive to CO2, one is sensitive to O2, get a bit of an overview of all these things, and then how all this information comes together in a central control. And as long as that central control is intact, then we have a very finely tuned system that ensures that we're getting as much air as we need. So it's rarely something, the good news is it's rarely something we have to worry about. Our bodies tend to maintain our respiratory parameters just fine. And so we can relax and do what we're doing and let our breathing take care of itself. Aside from that big breath before you start a long sentence. So with that, I'll end this long sentence, and hope that was interesting and useful, and we'll see you soon. Thanks for your attention.